Today, we're going to talk about the wonders and magic of meiosis. So meiosis has different phases. First, you have interphase. Interphase is the replication of a single sister chromatid, otherwise known as DNA replication. It is the resting phase between successive mitotic divisions of a cell, or it's between the first and second divisions of meiosis. It is preparation for meiosis. It is the first stage, stage uno, and during interphase, appropriate cellular components, such as DNA, are copied. Then you have prophase 1. Prophase 1 is when the chromosomes condense and the nuclear envelope breaks down, and then crossing over occurs. This is the exact reason why humans look unique, due to genetic recombination of DNA. Now, what is the nuclear envelope? The nuclear envelope surrounds the nucleus with a double membrane with multiple pores. The pores regulate the passage of macromolecules like protein and RNA, but they permit free passage of water, ions, ATP, and other small molecules. Then you have metaphase 1. Metaphase 1 is when pairs of homologous chromosomes move to the nucleus of the cell. They align on the metaphase plate in between the poles. Spindle fibers are fully formed. Then you have anaphase 1. Homologous chromosomes move to the opposite poles of the cell. Then you would have telophase 1 and cytokinesis. Chromosomes gather at the poles of the cells. The cytoplasm divides in cytokinesis. That is the main idea of cytokinesis. Then you have prophase 2. Prophase 2 is when a new spindle forms around the chromosomes. Spindle fibers form a protein structure that divides the genetic material in a cell. Then you have metaphase 2. Metaphase 2 is where the chromosomes line up at the equator, which is the center of the cell. Then you have the chromosomes become arranged on the metaphase plate, much as the chromosomes do in mitosis, and are attached to the now fully formed spindle. Each of the daughter cells complete the formation of a spindle apparatus. What is a spindle apparatus? The spindle apparatus refers to the cytoskeletal structure of eukaryotic cells that forms during cell division to separate sister chromatids between daughter cells. Then you have anaphase 2. Anaphase 2 is where centromeres divide and the sister chromatids separate due to the spindle fibers. Then you have chromatids moving to the opposite poles of the cell, the opposite sides. But then you have telophase 2 and cytokinesis. Now, telophase 2 is when the nuclear envelope forms around each set of chromosomes. Then the cytoplasm divides and the sister chromatids separate due to the spindle fibers. But overall, why am I telling you this information? Why is meiosis important? Well, meiosis is important because it ensures that all organisms produced via sexual reproduction contain the correct number of chromosomes. Meiosis also produces genetic variation by way of the process of recombination. You start with a diploid cell that produces 4 haploid cells that are genetically different from each other. Now, they also include many gametes, and meiosis is very important to gametes. What are gametes? Gametes are sex cells. Now, I hope you understood what meiosis is, why it's important, and I hope you have a great day.